So here's the cliffhanger that I left you at the end of our last exciting episode. One of these characters is telling the truth, the other is lying. How do you know the difference? Well, certainly not just by looking at the organisms. Okay? Um, Particularly with genetic data, if two organisms that are very, very distantly related happen to have the same mutation and the same adenine changes to a thymine at the same position in the DNA, you're never going to be able to tell by looking at the present day genetic sequences. However, there's a nifty principle that we can take advantage of to figure out which of these characters is telling the truth. And basically the way to sum it up is this. Different liars will tell different lies, but all truth tellers will sell the same truth. Okay, so if you've got different characters that lie, different homoplasies, they ought to all be telling different lies. Okay, but your synapomorphies, your true synapomorphies, all ought to tell the same story. They ought to tell you about the same evolutionary history. In other words, if you keep looking at more and more characters, what will happen is that all the synapomorphies will keep pointing toward the same correct evolutionary history. They'll keep supporting the same true phylogeny. The homoplasies will also start piling up, but each homoplasie that comes along will be pointing at a different wrong hypothesis. It'll be leading you toward a different wrong phylogeny. And eventually what happens is as you keep collecting characters, the synapomorphies, more and more of them start pointing toward the correct hypothesis, and they sort of outvote, they overwhelm all the different wrong lies that the homoplasies are telling you. Whew. So this is what we do. We look at what each character is telling us, and here they all are. Two species have the derived character state from mammary glands, so that this trait claims they belong together in a clade. Lack of fins put these six together in a clade. Fur puts these two together in a clade, and on and on and on. This one, we can ignore it. Goodbye, because it doesn't really tell us anything useful. But all of these other ones, as far as we can tell, each one on its own seems to be a synapomorphy. At least, we've got these shared derived character states for each of these remaining 8 characters. What do we do now? Well, the simplest thing to do is start at the bottom. Where is the biggest clade within this group? Because that's probably somewhere down near the root. And here it is. This is the biggest clade that we've got. So we're going to put that down at the bottom here, and then we're going to start nesting clades within it. Because remember that what you can do with clades is you can nest them within one another. If you can fit one clade entirely within another, they don't disagree. So for example, claws or nails here has the chimpanzee, the crocodile, mouse, pigeon, and lizard. That nests within this one. Okay, this clade lacks the salamander, but all the others could form a subclade within this clade. Let's see how that looks. So we start with lack of fins. That's the character that has the biggest, the synapomorphy that has the biggest clade. And what that maybe implies is, okay, when we put claws or nails in here, or lack of gills, they actually have the same, they have the same synapomorphy, they have the same synapomorphic group. What that implies is that all of these belong to one lineage, and that the salamander kind of split off on itself after this point in time. Okay, so those nest within one another. There's no disagreement there. So okay, we haven't found any problems yet. Well, okay, warm-bloodedness, yeah, okay, that does not disagree with this character. It would nest just fine in here. And mammary glands and fur, those two characters would both nest just fine within that. So, okay, we haven't found any problems yet. Let's see, carrot and scales, that nests just fine into here, and gizzard nests just fine into there. Okay, except there's one problem. Again, if the pigeon is most closely related to the crocodile, it cannot be most closely related to the mouse and the chimpanzee. So these two traits disagree. These two character states disagree. In the same way, if the pigeon is more closely related to the crocodile and the lizard than it is to the mouse and the chimpanzee, these two character states, or these two characters rather, disagree. At this point, what we do is we invoke this principle called parsimony. There are two possible explanations for what we're seeing here. Okay, one is that this is the true evolutionary history over here. Really, the pigeon is more closely related to the crocodile than it is to anything else. But after it split off from the crocodile, it evolved to be warm-blooded. And so that's why it ends up looking like it belongs in this group over here. The other possibility is this, that the pigeon really is the closest relative to the chimpanzee and the mouse, but after it split away from this lineage, it evolved a gizzard and it evolved carrot and scales, and so it looks like it belongs in this clade and this clade. Okay, I'm oversimplifying things a little bit here, but there are other ways in which you could get this pattern, but all of them basically involve the same thing. If this is a true statement about the evolutionary history of these organisms, then you had to have at least two evolutionary changes for the pigeon to end up in these two groups over here. Whereas if this is the true evolutionary history over here, you only had to have one coincidence, one homoplasy, to get the pigeon over here. Parsimony is a principle that suggests that, look, go with the simplest explanation. In this case, there were either two coincidences or one. The idea that these are both homoplasies requires two coincidences. The hypothesis that this is a homoplasie only requires one coincidence. And so we lean toward this being the correct hypothesis, and we toss out this trait as a homoplasie. I mean, in reality, when you've got a vote of 2 against 1, that's not really strong evidence that this is the correct hypothesis about the evolutionary history, and this is not. What you really want is lots and lots and lots of traits piling up on this side and very few over on this side. But just for the purpose of this demonstration, basically what I want to show you is that this is how essentially synapomorphies will end up outvoting homoplasies. Okay, once we throw out this character, do we have any more disagreements? Okay, once we decide that, okay, warm-bloodedness must have been a homoplasie, do we have any more disagreements? Okay, this nests within this nests in here, and this nests in here. Okay, we don't. So basically what you're seeing here is the true evolutionary tree. We've completely reconstructed the evolutionary history of this group of organisms. And let me show you how this works by walking through what we've reconstructed. Okay, so we've got this group here, then this group here, then it splits. This group goes off to one side, this group goes off the other side, and then we get the crocodile and the pigeon together. Let's see how that plays out through evolutionary time. What does this imply? Well, what do we know to begin with? We know that there's a lineage at the root of this tree that we're trying to reconstruct that leads to all 7 of these species. Okay, and so What's our first character, the biggest character, the one with the biggest, the largest number of the largest number of organisms that share the derived trait? It's lack of fins. Okay, well, lack of fins, let's see, that suggests that all 6 of these belong together in a clade. Oh, but it leaves out the perch. What that implies is that down here at the root of the tree, there was a split, and that the fish split one way, the lineage leading to the modern fish split one direction, and the lineage leading to these species went the other direction. Okay, our next character, the next largest clade that our characters are telling us about, is claws or nails. Lack of gills, by the way, is telling us about the same clade. It says that the chimpanzee, the crocodile, the mouse, the pigeon, and the lizard all belong together in a clade. Okay, so we look at that. Oh, okay, the salamander isn't part of either of those. What does that imply? Well, it implies that the lineage leading to all these species splits, and the salamander went one direction. Okay, one of these, after the speciation event, one of these lineages led to the modern day salamander, and the other one leads to all of these species. Next, we have 2 characters that tell us about the splitting of this group. One of these, you've got mammary glands and fur suggesting this clade, and you've got keratin scales suggesting this one. Well, that splits this group right in half. Suggests that this lineage here went through a speciation event. You have one daughter lineage that's going to lead to this clade, and another daughter lineage that's going to lead to this clade. Okay, there's our split. Here our organisms move apart. Okay, this is the last character we have to look at, gizzard. Suggests that the crocodile and the pigeon are one another's closest relatives. The lizard here gets left out. What that implies is that the lineage leading to that set of descendants splits. The lizard goes in one direction, and the 2 other organisms go in this direction. This is the lineage that evolves the gizzard. Okay, so we get this split. And that's it. That's all the characters we have. Well, we do have to do some more splitting here, but that's trivial because we know that these are all different species in the present day. We know that at some point in the past, they split apart from one another. And it's got to be basically between this point in time and the present day organisms. So here they go. We split them apart. And we follow through to what we see in today's world. So one character at a time, we split this clade away from that lineage, then this clade split away based on another 2 characters, then this group here split away from this one based on things like mammary glands and fur and keratinous scales. The presence of the gizzard implies that these 2 belong together, and so it split away from this. And then all the species separated, and here we are. We have completely reconstructed the phylogeny for this group. Ta-da! One thing that you should keep in mind is that whenever a phylogeny is reconstructed, it's a hypothesis. Okay, they're not facts. And as we gather more data, often phylogenies have to get shifted a little bit. We get more genetic data, and oh, okay, all of a sudden looks that species A is really most closely related to species B. At first, when we started getting in genetic data, it caused massive shifts in a lot of the evolutionary understandings that we thought we had. Because we were getting all of this new data, in some cases told us completely new things. We learned a lot about the tree of life once we start getting that data. As people gather more data and as computer algorithms can more powerfully crank this data to make these evolutionary trees, we find that we're adjusting the trees less and less. So we're homing in on the true tree, but we can never be 100% sure that we're there. If you can, you always want to double check a tree with other sets of data. So for example, if you've built a genetic or a tree that's based on morphology, that's physical characteristics, let's compare that to a phylogeny that's based on DNA sequences. Do they tell you the same thing? Well, yeah, pretty much they do. There are a couple of, there are one exception here. The genetic data say that the turtles belong here as the close relatives of the crocodiles and birds. That's not what the morphology says. Huh, I wonder what happened there. That's another mystery that poses another question to solve. But for the most part, you see that these two phylogenies support one another, and that's very encouraging. Once you've got a phylogeny that you feel really confident in, there's something neat that you can do. You can actually use the information that's present in the existing organisms, the present day organisms, and you can use that to go back in time and reconstruct what their ancestors were like. Here's the logic of how that goes. Let's take our vertebrates that you're very familiar with here. Oops. Let's take our vertebrates that you're really familiar with here, and let's look at mammary glands. All right. The chimpanzee and the mouse both have mammary glands. What does that imply about when that evolved? Well, it kind of suggests that this ancestor had mammary glands, but that this one didn't. So because if mammary glands evolved somewhere after this ancestor, you would expect only the mouse or only the chimpanzee to have them, right? So this was the latest that it could have evolved. If it had evolved somewhere back here, you'd expect all these species to have mammary glands too. So the most parsimonious reconstruction is that mammary glands evolved somewhere on this branch. Okay, you can go through and do that for any character that any of these species have. Okay? Another way of approaching it, you remember that one synapomorphy, like mammary glands, supports one clade. Here's the group of organisms that share the synapomorphy. This implies that you can stretch that box back past the most recent common ancestor for that clade. It implies that this ancestor here was the first to have that derived character state. And so the tick mark that denotes the evolution of that character state ought to go there. Generally, this is how we denote evolutionary changes on a phylogeny. You do a little tick mark across the line that says, hey, this is the branch where we think this trait evolved, and you jot the name of the derived character state down below that or above that. There are different ways of writing it, but they basically all come out to the same thing. You're labeling the point on the tree where you think a particular character state evolved. And this is something that you could be asked to do on, for example, an exam. Remember that when you're doing a homoplasy, you have to put in multiple state changes. So for example, with a pigeon, you need to show that warm-bloodedness evolved here and that it evolved there. Okay, so we can go ahead and add these tick marks for all of the characters that we've been looking at. In fact, we could add tick marks for any character that is in any of these organisms in this group, because we can reconstruct where along this phylogeny those traits evolved, assuming that we really trust this phylogeny. Once you've got those, you can reconstruct for these particular characters what any of these ancestors looked like. So for example, here's an ancestor you might be interested in. This is the ancestor of all amniotes. One of the really important evolutionary leaps forward among vertebrates was the evolution of what's called the amniotic egg. This gave animals, really for the first time, gave vertebrates for the first time the ability to reproduce away from water. It allowed these organisms, the reptiles and the mammals, to essentially colonize dry land and really take over in the niches where we see them today. Okay, but what did this ancestor look like? Let's suppose we don't have a fossil record of that ancestor. Can we reconstruct what its morphology was like? Well, yeah, up to a point. I mean, presumably that lineage that led to this point, started out looking like the outgroup, the lamprey, and then we just walk forward from the root to this node. So we're kind of going along here. It's evolving. Oh, okay. So we've got a lamprey, except it lost its fins. So, all right, we've got an organism without a fin. We're going to keep going here. Oh, it lost the gills too, and it got claws. So that's what we've got. It's not a lamprey anymore, but whatever the ancestor to all the amniotes was, it didn't have mammary glands or fur, it wasn't warm-blooded, didn't go through metamorphosis or have keratin scales or gizzard, but it did have claws or nails. It didn't have fins, and it didn't have gills. Okay, this may seem kind of trivial, that we can reconstruct a few characters for whatever this ancestor was here. But when you get a lot of characters and a lot of species, you can do much more powerful things with this approach. So, for example, a study that came out a few years ago, what scientists did is they took the sequence for the rhodopsin gene in living reptiles, including birds. And they reconstructed the genetic sequence. Actually, I think it was probably the protein sequence, but it was either genetic or protein sequence for the rhodopsin gene for dinosaurs. Okay, so they knew what the sequence was for birds, they knew what it was for crocodiles, they knew what it was for lizards and snakes and tuataras, and they used all that to say, okay, this is the most probable sequence for this protein in dinosaurs. What was neat is that they then put together that protein, and they actually, I think what they did is they cloned that gene into a cell line and got that cell line to express this particular variant of the rhodopsin gene. And they took it into the lab and they exposed it to different wavelengths of light and saw what it reacted to. And what they found is that this rhodopsin molecule is most sensitive under low light conditions. The implication being that dinosaurs could see in the dark. Okay, so what scientists basically did is from the information in living day organisms, they reconstructed a gene that has not existed for millions of years. And from the behavior of the protein that's made by that gene, they were able to infer something important about the habit of organisms that have never been seen by any living human. As we get more and more genetic data, we're going to be able to do more and more of this. In fact, we're shortly going to be getting to the point where we will be able to reconstruct the entire genomes of organisms that have been extinct for a very long time. And what this could do is it could open up all kinds of exciting doors to our understanding of how the organisms that we see on Earth today have evolved.